Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. Before we jump in today, please do remember to like this video and subscribe to the Sand Science Experience. It really does help support what we do here. Okay, so today we're plunging into some truly groundbreaking stuff, a study that's kind of flipping our understanding of how our bodies renew themselves, right down at the cellular level. We're talking about something we all have, but let's be honest, rarely think about. The age of our little cellular powerhouses, the mitochondria, and how their age plays this surprisingly pivotal role in deciding the fate of crucial stem cells. This isn't just about cells getting old and worn out. No, this deep dive is going to reveal how old, in this context, can actually be a really good thing, like leading to unexpected regenerative potential, especially as we ourselves get older. It's kind of a hidden wisdom in our own biology.
Absolutely. And our mission today is really to unpack this fascinating paper that just came out in Nature Metabolism. The core problem it tackles is, well, it's a big one. How do our tissues actually maintain themselves? Constantly repairing, replacing cells throughout life. And what exactly goes wrong in that process as we age? We've known for a while that cellular metabolism is a key regulator. It influences what a cell becomes, what it does, but the specific role of organelle age, like the age of those tiny mitochondria inside the cell in determining cell fate, that's been pretty much a mystery, a black box. Until now. So this study zooms right in on intestinal stem cells, ISCs, and their crucial support cells, the Paneth cells, or PCs. And what they found is this previously unrecognized diversity, both metabolic and functional diversity, driven fundamentally by how old their mitochondria are. So yeah, our mission for this deep dive is to peel back those layers, understand how these old mitochondria influence stem cells, and maybe most importantly, how this could open up new doors for regenerative medicine, especially for aging.
Okay, let's get into it. This sounds really counterintuitive already. Researchers found something pretty astonishing in the small intestine stem cells. So it turns out, not all intestinal stem cells are the same. And the key difference isn't something obvious. It's actually the age of their mitochondria. Usually old means decline, right? But this study seems to say otherwise from the get-go. What's so special about these old cells?
That's exactly where the surprise is. What's really fascinating is this concept called asymmetric cell division. So imagine a stem cell dividing. It's not just about the two new daughter cells, but about what stuff each one inherits from the parent. And in these mouse intestinal stem cells, they found a specific group of daughter cells. They called them ISC-MitoO for ISCs with old mitochondria that specifically get loaded up with the old mitochondria.
Right, the old mitochondria. How do they even tell old from young?
Yeah, that was clever. They use this temporal labeling system in a special mouse model. Think of it like putting a timestamp on the mitochondria. They use a genetic tag. Mitochondria hang around for 48 hours, got the old label. Newly made ones, tagged within just 8 hours, were young. Super precise tracking. And these ISC-MitoO cells, they found them mostly in the upper part of the stem cell niche, usually just one or two per little crypt, those folds in the intestine lining. So a small group, maybe about 9% of all the ISCs, but distinct.
Okay, so we've got these ISC-MitoO cells set apart just by having older mitochondria. Wild. But functionally, what does it mean? Do they act differently than the ones with young mitochondria, the ISC-MitoY? What's the payoff for having these older engines?
Right. That's the critical question. What's the functional consequence? So they tested them in the lab, in vitro, looked at their ability to form organoids.
Meaty guts in a dish, basically.
Exactly. These little self-organizing structures that mimic the real thing. And here's the kicker. The ISC-MitoO cells, the ones with old mitochondria, were way better at forming these organoids. And crucially, they could do it independently of their usual support cells, the Paneth cell niche.
Independently? That sounds as a big deal.
It is a huge deal. Normally, stem cells really need those support cells sending signals, but these ISC-MitoO cells were so potent, they could actually recreate that Paneth cell niche all by themselves. A clear advantage over the ISC-MitoY cells, the ones with young mitochondria, which kind of struggled without that external help.
Wow.
And just to be clear, these aren't those slow-cycling reserve stem cells we sometimes hear about. No, these ISC-MitoO cells divide at similar rates. Their special power really comes down to the mitochondrial age itself. It totally turns the usual thinking about cell aging on its head.
It really does. Older mitochondria giving this massive regenerative advantage.
Yeah.
It makes you think, okay, how? How do these old mitochondria give them this superpower?
Yeah.
Is it just being old, or is there some specific metabolic trick going on? This is where it gets really interesting, right? The mechanism.
Absolutely. It's all about the metabolism. And it's quite elegant, but also a bit paradoxical. When they looked closely at the metabolism, they found that while the mitochondria had slightly lower cristae density, those internal folds,
Which usually means less energy production capacity.
Typically, yeah. You'd think less surface area for oxidative phosphorylation. But here's the paradox. Despite that, their key metabolic difference was a much higher level of a metabolite called alpha-ketoglutarate, or AKG.
Higher AKG, even with potentially less efficient mitochondria. How does that work?
It suggests maybe their mitochondria are sort of uncoupling things, prioritizing something other than just raw ATP production. And tracing experiments actually showed the TCA cycle, the main energy pathway, was running faster in these ISC-mito cells, leading to this buildup of AKG.
Okay, so AKG pops up as the potential master key. For those of us maybe not deep in biochemistry, what is alpha-ketoglutarate? What does this stuff actually do in the cell to give it these powers? How does it connect back to rebuilding that Paneth cell niche?
Great question. So AKG, alpha-ketoglutarate, it's a critical epigenetic cofactor.
Epigenetic. Like controlling which genes are switched on or off?
Exactly. Think of DNA as the instruction manual. Epigenetics are like sticky notes telling the cell which instructions to read, when, and how loudly, without changing the actual text. AKG helps certain enzymes place or remove these sticky notes. It acts as fuel for them, basically. So they did this key experiment. They took the ISC-mito Y cells, the ones with young mitochondria, and fed them a form of AKG that can get into cells easily, DMAKG. And the results, pretty stunning. It boosted their ability to form organoids independently, almost up to the level of the naturally powerful ISC-mito O cells.
Wow.
And even more importantly, it directly promoted their ability to make Paneth cells to recreate that niche. Direct proof that AKG pushes cells toward that Paneth cell fate.
So how does AKG do that? Which enzymes are involved?
It connects to a family of enzymes called TE enzymes, 10-11 translocation enzymes. These enzymes use AKG to modify DNA, specifically creating a mark called 5-hydroxymethylcytosine, or 5-HAMSC. And guess what? ISC-mito cells naturally had higher levels of this 5-HAMAC mark.
Ah, okay.
Right. And when they blocked the TET enzymes, the effect of adding DMAKG was lost. It also reduced the ability of the AHENO cells to make Paneth cells, so a clear chain. AKG fuels TE enzymes, TA others make 5-AKC marks, and this promotes Paneth cell formation. They even found it's about balance. AKG boosts TAT, but another metabolite, 2-HG, inhibits it. ISC-mito had less 2-HG, tipping the balance towards TET8 activity.
And did they pinpoint a specific TE enzyme?
They did, using CRISPR. Knocking out TET2 specifically abolished the effect of AKG. So TET2 seems to be the crucial player in this pathway. It's this really neat metabolic control switch for cell identity.
That is genuinely mind-bending. A single metabolite, AKG, acting like a switch to reprogram cell fate through epigenetics. But, okay, the big question always is, does this work outside the lab dish? In a whole living animal, what's the bigger picture, especially thinking about aging? Can this actually help aged tissues?
Yeah, connecting it to the bigger picture, the in vivo results are profound and very relevant to aging. They gave DMAKG to mice, and yes, it boosted AKG levels in the gut lining, and importantly, it increased that key 5-epigenetic mark in their intestinal stem cells, and functionally, it directly sped up the turnover and renewal of Paneth cells.
Okay, why is Paneth cell renewal so important for aging?
Because intestinal renewal slows down as we age, that's pretty well known, but paradoxically, older individuals can actually have more Paneth cells. The problem is these aged Paneth cells often start pumping out this molecule called Notum.
Notum.
Yeah, it's a Want pathway inhibitor. Want signaling is crucial for stem cell function and regeneration, so Notum basically puts the brakes on repair. It's like having more workers, but they're actively sabotaging the work.
Ah, okay, so getting rid of those sabotage cells would be good.
Exactly. And this is what's so exciting. Treating aged animals with DMAKG increased Paneth cell renewal. And the kicker, the newly made Paneth cells had much lower levels of Notum expression. So it's like you're metabolically flushing out the defective, inhibitory old cells and replacing them with fresh, functional new ones that don't block regeneration. It's not just renewal, it's better renewal.
Better renewal, especially in older systems. That is a potential game changer. Okay, but what about actual damage? Like, say, after chemotherapy, which is notoriously hard on the gut, especially in older patients, can DMAKG help recovery then?
Absolutely. That's the crucial test for potential therapy, right? Can it help recover from real injury? So they tested this. They pre-treated aged mice with DMAKG before giving them 5-fluorouracil, or 5-FU, a chemo drug that really damages the intestinal lining and causes weight loss.
A tough challenge for an old gut.
A very tough challenge, and the results were, well, really striking. The DMAKG pretreatment significantly reduced the weight loss in the aged mice. That's a direct sign of better gut function. And importantly, it improved the recovery of the gut lining itself, the epithelial cell density, bringing it back up to a rate that was comparable to young animals recovering from the same damage.
Wow, so it essentially helps the aged gut bounce back like a young one.
Pretty much. It's strong proof of principle that you can metabolically target specific aged cell types, promote their replacement in vivo, and significantly improve tissue regeneration and recovery from injury, even in an aged system. That's powerful.
So what does this all mean for you listening? Well, I think this deep dive is a fantastic reminder that even tiny things inside our cells, like how old our mitochondria are, can hold these really profound, sometimes counterintuitive secrets about our health and our ability to regenerate. This research clearly points towards a potential future where targeting specific metabolic pathways, maybe using molecules like alpha-ketoglutarate, could become a really powerful tool, helping us fight age-related decline and repair tissues better.
Yeah, our discovery really highlights that mitochondrial age isn't just a passive marker. It actively creates functional diversity among stem cells.
Right.
It sets up this metabolic bias that it turns out we can actually harness. It underscores just how active and crucial cellular metabolism is in deciding what cells become and what they do.
Right.
And that opens up some really exciting possibilities for, therapeutically guiding stem cells to do a better job, especially as we get older.
Which leaves us with a pretty big thought to chew on, doesn't it? Could understanding and maybe even manipulating the age of our organelles and specific metabolites like AKG, could this be a kind of universal strategy? A way to combat age-related decline and boost regeneration not just in the gut, but maybe across all our tissues where stem cells falter with age. Definitely something to mull over until our next deep dive. If you enjoyed this deep dive into the fascinating world of cellular renewal and mitochondrial age, please do remember to like this video and subscribe to the SANS Science Experience. It really helps us keep bringing you the latest breakthroughs. Thanks for joining us.